Welcome back to Biology 2225 at CNM. We are talking about the urinary system, which is chapter 23 in your textbooks. We've gotten ourselves up through objective 37, so we've done most of the job of describing the structure of a nephron. All that's left for this section is to really finish up by contrasting the juxtamedullary and cortical nephrons and talking a little bit about the nerves that are associated with nephrons. Then we'll be able to begin talking about the physiology of urine production. So what we see in objective 38 is that there's two types of nephrons in a kidney. We have what are called cortical nephrons and juxtamedullary nephrons. Cortical nephrons are by far the most common type of nephron, constituting about 85% of all nephrons. Cortical nephrons sit primarily in the cortex of the kidney. That's where the name cortical comes from. And most of them sit just beneath the renal capsule, so they tend to be very peripheral, near the edge of the parenchyma tissue. They have relatively short loops of Henle, or nephron loops, and in many, the loops only slightly dip down into the medulla of the kidney. In some cases, they don't dip into the medulla at all. It's even possible that in some cortical nephrons, there are no loops of Henle at all, though that's not the typical condition. These nephrons perform all the typical functions of nephrons in that they perform filtration followed by reabsorption and secretion. The second capillary bed in cortical nephrons is the peritubular capillary bed that we find investing the proximal convoluted tubule and distal convoluted tubule. Now, the less common type of nephron is called the juxtamedullary nephron. Juxta means near or next to, medullary, of course, meaning medulla. So these, again, are less common, only 15% or so of the nephrons, and just by virtue of their name, of course, they are farther away from the periphery of the kidney and deeper in the cortex, sitting primarily very near to the cortex medulla boundary. Juxtamedullary nephrons have extremely long loops of Henle, and as we'll see, these long loops of Henle are important in creating a concentration gradient of salt in the medulla. They create a very clear-cut salinity gradient, salt gradient in the medulla, which we'll touch on later. And again, these long loops of Henle, as I've hinted at earlier, are involved in conservation of water. In other words, reclaiming water back into the bloodstream. It's interesting to note that loops of Henle in various animals, mammals especially, correlate with the environment. And so some desert animals, for instance, are characterized by having extremely long loops of Henle, things like kangaroo rats, which live in the desert. And kangaroo rats rarely drink, getting most of their water from their food because they're so efficient at conserving water and produce a very low volume but highly concentrated urine. And again, that's because of the habitat in which they live. So these long loops of Henle clearly seem to be involved in conservation of water, or reabsorption of water back into the bloodstream. In the case of juxtamedullary nephrons, their second capillary bed is not the typical peritubular capillaries, but the specialized capillary bed known as the vasa recta, which we saw dive down into the medulla with the long loop of Henle. Here we see a diagram of the two distinct types of nephrons that we just discussed. Again, cortical nephrons, the most common, and again, the glomerulus or the cortical nephrons tend to be near the surface of the kidney. Loops of Henle barely diving down into the medulla, if at all. Juxtamedullary nephrons, of course, deeper The glomerulus resides deeper in the cortex, and the loop, very long loop of Henle dives deep into the medulla. And then finally, for this section of the chapter, we just want to talk a little bit about the innervation of the kidney. We have a so-called renal plexus. Remember, plexus is a network or a net-like branching pattern of either blood vessels or nerves, in this case nerves. And so we have a network, a branching network of nerves and ganglia, which, remember, ganglia are neuron cell bodies out in the peripheral nervous system, usually associated with the autonomic nervous system. And these are wrapped around the renal arteries. And the nerves then follow the renal artery into the tissue of the kidney. And the nerve fibers, once they get into the parenchyma, will continue along the blood vessels and also travel to the tubules, convoluted tubules of the nephron. Again, because these ganglia are often associated with the autonomic nervous system, what we find primarily in this innervation is sympathetic innervation from, of course, the sympathetic nervous system. As we will see later in the chapter, their sympathetic activity tends to reduce glomerular blood flow and the rate of urine production. And so again, during exercise, remember, we tend to shunt blood towards the muscles, and we reduce blood flow and activity of the kidneys just to conserve resources and send most of our resources to the muscular system. And so these autonomic fibers are important, as we'll see, in reducing glomerular filtration and glomerular blood flow. Also, they become part of blood pressure regulation. When blood pressure falls, these sympathetic fibers can stimulate the kidneys to secrete renin. And you may recall from a previous chapter when we discussed blood pressure, renin leads to the production of angiotensin II, which then, through a variety of effects, causes an increase in blood pressure. We're actually going to come back to this when we talk about regulation of glomerular filtration rates. It is interesting to note that the kidneys do also receive some parasympathetic innervation through this renal plexus, but it is still unclear exactly what the function is of this parasympathetic innervation. Okay, so we are ready now to talk about the physiology involved in the formation of urine. And in section 3, we begin by talking about filtration. Remember, there are going to be three basically different physiological processes occurring in the formation of urine. We begin with filtration, and again, just to recap, this happens at the glomerulus. And recall that filtration is the movement of materials out of the blood into the tissues. And the tissues in this case is the hollow glomerular capsule. And so we're going to spend quite a bit of time talking about the dynamics of filtration and the various ways that we can control glomerular filtration rates. As we will see, we filter in all of our glomeruli combined, we filter about 180 liters of fluid a day from the blood into the glomerular capsule. Now, clearly, we can't afford to lose 180 liters of fluid a day. I mean, if you think back to our first chapter, we only have about 3 liters of blood plasma to work with in the entire body. So clearly, 180 liters is a very high volume that we could never afford to lose in the form of urine. And so what has to happen following filtration is we have to perform a second physiological function known as tubular reabsorption. Tubular reabsorption, recall, is the opposite of filtration, where we move materials back into the bloodstream from the nephron. And this is a very important process in reclaiming materials back into the blood. And so as it turns out, and as we'll see, about 99% of what we filter actually gets reabsorbed back into the blood. But it's a very selective reabsorption, and so the 1% or so, it's actually a little less than 1%, that remains in the tubes is just the materials that we want to get rid of, the materials we want to excrete. So remember, filtration is fairly nonspecific. There are some things like size that will determine what gets filtered and what doesn't, but it's a relatively nonspecific process. Reabsorption is very specific, and this really determines what's going to be left in the tubules and ultimately become urine that we secrete. And then finally, there is a third process that we've touched on called tubular secretion. In secretion, we do essentially movement of materials in the same direction as filtration, which means once again we're going to take materials from the blood, from the plasma, and move it into the tubules. But this time it's going to be very selective. Unlike filtration, which really only filtered things based on size, tubular secretion is going to use transport molecules in a very specific way to fine-tune the contents of the tubular fluid. And so this is how we get rid of things that we might not have been able to filter properly in the first place. So for instance, as blood flows through the glomerulus, there may be something, some material, some toxin, or some waste product that we want to completely remove from the bloodstream. But through filtration, we might only be able to remove, say, 80% of it, and a certain amount remains behind. Well, as the blood flows into the second capillary bed, that remaining 20% we may be able to secrete into the tubules, and that's how we might be able to accomplish ridding the blood of 100% of something as opposed to just 80% of something. It's also possible that there might be some things in the blood that are simply too big to filter, but we still want to get rid of them, and so secretion is a selective process that we can use to selectively get rid of those materials that we either couldn't filter or couldn't filter enough in the first place. The book also lists a fourth process here called water conservation. In my way of thinking, water conservation is just a specific example of tubular reabsorption. It's just that specifically what we're reabsorbing back into the tissues and specifically back into the blood is water. So I don't think of that as a fourth separate physiological process. I just think of it as a type of tubular reabsorption. So again, in this section 3 of the textbook, we're going to focus for now on glomerular filtration and what determines how much filtration occurs and how we regulate that. All right, so the fluid that comes out of the blood plasma during filtration and winds up in the glomerular capsule is referred to as glomerular filtrate, or just simply filtrate. We only call it filtrate while the fluid's in the capsular space of the glomerular capsule. By the time it moves on to the proximal convoluted tubule, we then refer to it as tubular fluid. Tubular fluid, and we call it tubular fluid all the way from the proximal convoluted tubule into the loop of Henle and on through to the distal convoluted tubule. And of course, during this process, during this movement through the nephron, substances are being removed and added through the processes of reabsorption and secretion. By the time we get to the collecting duct, when the distal convoluted tubule dumps into the collecting duct, at that point, we do call the fluid urine at that point, even though the processing might not be completely finished. Beyond this point, really, the only change significant change that might take place is, again, reabsorption of water, the very last final control to determine exactly how concentrated the urine might be in terms of solute concentration. But other than that, it doesn't change very much, so we do call it urine once it makes it into the collecting duct. So just a recap of the glomerulus. Again, you have a glomerular capsule with a outer parietal layer of squamous cells, and then an inner form-fitting layer that covers the glomerulus with the podocytes, the so-called podocytes. And these podocytes have extensions Remember, they have these interlocking extensions. Those extensions, by the way, are referred to as pedicels. And the spaces between the pedicels of podocytes are called, remember, filtration slits. Just a quick recap of the basic anatomy. So remember, glomerular filtration is like filtration in any capillary, in the sense that hydrostatic pressure, blood pressure, is the primary force that tends to push fluid out. But as we'll see in a few minutes, there are some differences in terms of the numbers, the actual numbers of hydrostatic pressure and osmotic pressure that govern filtration. But it is the movement of materials from blood into the tissue space, or in this case, the glomerular capsule. For materials to move, of course, they have to pass through some kind of barrier. And this barrier between the blood and the glomerulus and the capsular space is referred to as the filtration membrane, somewhat reminiscent of the respiratory membrane in alveoli, which allows gases to exchange between blood and air. Here we're talking about the exchange of dissolved molecules between blood plasma and fluid filtrate in the capsular space. So what are the layers of this membrane? Again, somewhat reminiscent of the respiratory membrane. There are 3 layers. There are 2 cell layers and a shared basement lamina, or basement membrane, between them. The first layer, again, like the respiratory membrane, is the squamous epithelium of the capillary, the glomerulus. Then a shared basement membrane that anchors the 2 epithelial layers to each other. And then on the capsular side, of course, as we said, the inner lining of the capsule, which consists again of the podocytes with their foot processes, or pedicels, interlocking with one another. Important to recall that the capillary bed that we have in the glomerulus is a fenestrated capillary, one of the more porous types of capillaries, because we want to encourage movement of materials. And so a fenestrated capillary is a capillary of choice. Remember, fenestrated capillaries have large pores in them of about 70 to 90 nanometers in diameter, and so these are highly permeable capillaries, and most of the diffusion passes through the pores. These pores, though, as you can see by this to scale diagram, are not large enough, generally speaking, to allow any blood cells to pass through. So if you do find blood cells in the urine, it's often indicative of some kind of damage to the filtration membrane, which doesn't normally allow blood cells to pass through. So what kinds of things do pass through? Essentially, anything that's under 8 nanometers in diameter. Again, this filtration is pretty much based on size. And so large molecules greater than 8 nanometers tend not to be able to make that through. So what does that include? Well, protein-bound minerals and hormones. So remember, certain minerals and things like iron that are ferried around the blood by protein carriers like albumins and so on will not be able to make it through because of the size of their protein carriers. Anionic compounds, especially large anionic compounds, tend not to be able to make it through. Again, most of the plasma proteins, and as I said earlier, blood cells, all these things tend not to make it through because they're too big to pass through these fenestrations. The basement membrane that anchors the 2 cell layers together is essentially a proteoglycan gelatinous material. Remember, proteoglycan means a protein carbohydrate complex. And this also gets involved in determining what gets filtered. So there are some molecules that are small enough to pass through the filtration slits, but they, because again, the filtration slits or the filtration pores, the fenestrations in the capillary are 70 to 90 nanometers in diameter, but we see that anything greater than 8 nanometers starts to have difficulty. So clearly they're small enough to get through the fenestrations. What they have difficulty getting through is this proteoglycan gel which is the, again, basement membrane. And then finally, again, we have podocytes on the capsular side of the filtration membrane. And as I described earlier, podocytes are octopus-like or star-shaped with many projections. And each of these projections is called a foot process or a pedicel. And it's these pedicels that tend to wrap around the capillaries, again with their jagged edges that tend to interlock with one another, almost forming what looks like the serrated sutures of the skull. Here we just call them interdigitating pedicels or foot processes of the podocytes. So this creates tiny spaces between these pedicels that are interlocking with one another, and these tiny gaps between the adjacent pedicels are referred to as the filtration slits. The filtration slits are about 30 nanometers wide, and so once again they constitute an obstacle to certain sized particles trying to get through, especially large anions again. And so, in summary, what tends the kind of filtration that takes place is, as we see in Objective 52, most molecules, regardless of charge, if they're smaller than 3 nanometers, tend to be able to pass through this filtration membrane and into the capsular space. Substances that are of low molecular weight and a little bit larger than 3 nanometers can make it through, but they don't make it through quite as easily. So what are the small kinds of molecules that can make it through, pass through readily? And we see that listed here. And you'll notice most of these are essentially tiny molecules. Water, of course, H2O, only 3 atoms. Electrolytes, right? Single ions like sodium, potassium, chloride, magnesium, calcium, etc. Glucose, a small building block of carbohydrates. Amino acids, a small building block of proteins. Fatty acids, some of the larger molecules are able to pass through, because remember, fatty acids can be fairly long chains of carbon and hydrogen, but they don't have great diameter. They tend to be more linear, so they pass through. Small vitamins, urea, of course, we want to pass through. That's a waste product that we're trying to rid the blood of, as well as uric acid and creatinine. These are the nitrogenous wastes that we find in the urine, so it's no surprise that they pass through easily. So this process of filtration is referred to as glomerular filtration, and as I said earlier, it's just a special case of exchange between blood plasma and the surrounding tissues. In this case, the tissue is the capsular space. I've already described the membrane and its 3 components. So here you see in written words what I was just describing on that previous diagram. Here we see an electron micrograph of podocytes covering the capillaries of a glomerulus, and again these star-shaped or octopus-shaped podocyte with its pedicels, right? Its pedicels, these extensions of the cells, and the filtration slits are all these very narrow gaps or spaces between the interlocking arms of the pedicels. And you see how the podocytes really crawl over the entire surface of the glomerulus, helping contribute to the filtration process. So as I said earlier, almost any molecule smaller than 3 nanometers can pass freely, and that was that list of things. And then we said substances that you might guess that would be small enough to pass through, but are carried on carrier proteins, of course, don't make it through. If we can unbind them from their carrier proteins, then they will pass through freely. And these are things like calcium, iron, and thyroid hormone, all of which are usually transported via some kind of carrier like transferrin for iron. As I said, normally the filtration membrane doesn't allow things like cells to pass through or large molecules like proteins. And so if there is damage to the filtration membrane, sometimes you will see these things show up in the urine, right? Because whatever passes into the through the filtration membrane and becomes filtrate ultimately becomes urine if it's not reabsorbed. And so if these things do end up in the urine, it's usually an indicator of some problem with the filtration membrane. So proteinuria or albuminuria is a name we give to the presence of protein in the urine, which is not normal. And hematuria is the name we use for the presence of blood in the urine, or blood cells specifically in the urine, and that also is not normal. Sometimes you see temporary episodes of proteinuria or hematuria in endurance type athletes because of the strenuous exercise. They send so much blood to their muscles for such a long period of time that the kidneys experience a prolonged episodes of perfusion. Apologize for the typo there, perfusion. And with that reduced blood flow for such a long period of time, say like during a marathon, the glomerulus actually begins to deteriorate because of the hypoxic conditions that occur during that 2 to 3 to 4 hours of, say, a marathon. And that deterioration can then lead to damage of the membrane, which leads to proteinuria or hematuria. Thankfully, this is temporary, and the glomeruli can reestablish themselves fairly quickly once the endurance activity is ended. All right, so we've been talking about filtration as the same kind of basic event that happens at every capillary bed, where, remember, we tend to filter more fluid out than we reclaim by reabsorption, which leads to a buildup of fluids in the tissues, and that's why we needed a lymphatic system to drain away that excess fluid. So the basic dynamics are the same, but as I mentioned earlier, the numbers are quite different, and that does change things a little bit compared to a conventional capillary bed. So, as is with all capillary beds, the actual filtration pressure, the actual force that filters material out of the glomerulus, depends once again on the hydrostatic blood pressure within the capillary relative to the pressure outside the capillary, and also the differences in osmotic pressure between the blood and the filtrate in the capsule. So same considerations as before, but let's take a look at the numbers, the actual numbers. Let's start with hydrostatic pressure. In a conventional capillary, remember two things. We saw blood pressures ranging from 10 to 15 millimeters of mercury in most capillaries of the body. What we also saw in conventional capillaries is that pressure tended to drop between the proximal end of the capillary, the front end of the capillary or arterial end, and the venual end. So the pressure would drop significantly as the blood flowed through the capillary. When we look at a glomerulus, we see some things that are different. First of all, the pressure in the glomerulus is much higher than in a typical capillary bed. And we've talked about this previously in this chapter. How much higher? Well, you see here about 60 millimeters of mercury compared to the 10 or 15 that's in most capillary beds. And again, there are a couple of reasons for this high blood pressure. The afferent arteriole being larger than the efferent arteriole means you have a bigger inlet of blood, more blood coming in, and you get sort of a bottleneck effect as it tries to exit. That creates back pressure. Also remember, the second reason is that because the glomerulus is drained by an arteriole and not a venule, that also gives us the capacity to build up greater back pressure because the vessel, the draining vessel, doesn't expand as readily as a venule would. The other difference that I want to point out here, it's on a later slide, but I'm just going to mention it now, is that the blood pressure in the glomerulus does not tend to fall as much as we move through the glomerulus. So at the afferent arterial end, the pressure does not fall significantly as it moves through to the efferent arterial. That pressure tends to be maintained throughout the length of the glomerulus. And again, it's for these same two reasons: the bottleneck caused by the different sizes of the two vessels, and the fact that the efferent arterial is a resilient vessel that doesn't expand when it accepts blood. Now, we have to, in order to understand the net pressure that's going to push fluids out or materials out, we need to know what the pressure is outside the glomerulus, just like we had to know that pressure for conventional tissue spaces. Well, there is a hydrostatic pressure in the capsular space. It does fluctuate, but on average, the hydrostatic pressure in the capsular space outside the glomerulus is about 18 millimeters of mercury. And again, this pressure occurs due to the constant filtration of fluid into the capsular space, and it can only move so quickly out of the capsule into the proximal convoluted tubule. So there's a bottleneck at the proximal convoluted tubule, at the tubular pole, and that causes a little bit of back pressure to build up, which averages about 18 millimeters of mercury. So those are the numbers associated with the hydrostatic pressure. Now we have to consider the osmotic pressure conditions as well. Remember, osmotic pressure is a measure of solute concentration, essentially. What we basically have here is that the osmotic pressure of the blood is about the same in the glomerulus as it is everywhere else. So we're going to use basically the same number we used in our conventional conversation about capillaries, about 32 millimeters of mercury. Remember, this is a force that tends to draw fluid in. Water flow tends to move towards a higher osmotic pressure. So we've got a pressure of 32 due to the concentration of solutes in the blood that tends to draw water into the blood. Now, what about the filtrate? Well, the filtrate is relatively protein-free, as we've seen, and so it doesn't have a significant colloid osmotic pressure. Colloid osmotic pressure specifically refers to the concentration of proteins, and that is the most significant osmotic pressure that's going to dictate movement of water. And so for the osmotic pressure of the capsule, the filtrate in the capsule, we're going to give that a number of essentially 0. So when we add all these pressures up together, we're going to get something known as the net filtration pressure. Thinking back to what we've said already, we've got an outward pressure of 60 millimeters of mercury. That's the hydrostatic pressure in the glomerulus pushing outward. And that doesn't significantly change again as we move from the front to the back end of the glomerulus. Well, then we've got a hydrostatic pressure in the capsule of 18 millimeters. That, of course, is a pressure that's going to want to push fluid back into the blood. And we've got a pressure in the blood, colloid osmotic pressure in the blood of 32 that tends to want to draw fluid into the blood. So we put these all together. We've got a force of 60 millimeters of mercury pushing fluid out. We've got the hydrostatic pressure in the capsule of 18 pushing fluid back in. And we've got a pressure, an osmotic pressure in the blood itself of 32 drawing fluid into the blood. And so we subtract these two from the 60, the force that's pushing out, and we end up with a net pressure of 10 throughout the entire length of the glomerulus, net pressure of 10 out. That's the important thing to appreciate. The overall net pressure is a pressure out. And this is what we refer to as the net filtration pressure. A net pressure of 10 pushing materials out of the glomerulus. This net pressure of 10 is a little bit higher than the typical filtration pressure of a capillary bed. And probably the most important thing to realize is that this net pressure out does not drop throughout the length of the glomerulus. So it's about 10 at the front end of the glomerulus, and it's still about a net pressure of 10 at the back end of the glomerulus. Remember, in the conventional capillary, we have a net filtration pressure pushing out at the front end of a capillary bed, but a net reabsorption pressure at the back end of a capillary that's going to pull some of that filtrate back in. That does not happen in the glomerulus. There is no reabsorption back into the glomerulus. We only have a net force of 10 pushing out the entire length of the glomerulus. And that's really a major difference between this capillary bed and other capillary beds of the body. There is no reabsorption back in to the glomerulus. So all we have is filtration. We've already described why the blood pressure in the glomerulus is higher, and we see now why that's important. To combat these forces that would cause fluids to move back in, we need a high pressure to push materials out and get filtration, the level of filtration we need. Because of this high blood pressure, of course, the kidneys, the glomeruli specifically, are vulnerable to hypertension. The pressure is already high, and so if we have high systemic pressure, that can lead to even higher pressure in the glomeruli, higher than 60, and that can lead to glomerular rupture. And if that happens, of course, then we get scarring of the kidney tissue. This is known as nephrosclerosis, or sclerosis of the kidneys. And this replaces functional glomeruli with non-functional scar tissue. It can also lead to atherosclerosis of renal blood vessels. You may recall we said that high blood pressure tends to correlate with the buildup of plaque on the inside of blood vessels. That's atherosclerosis. And if that occurs significantly, we usually, of course, worry about that in terms of causing a heart attack or a stroke. But if it happens in the kidneys, of course, if we cut off blood flow to the kidneys, that's not only going to reduce the efficiency of the kidneys, but it will also lead to hypoxia and tissue death, and that can ultimately lead to renal failure. So yet another problem associated with atherosclerosis and high blood pressure, in addition to heart attack and stroke, is the potential for ultimately renal failure. So here we see a diagram of the forces that we have just been discussing. Again, the yellow is showing the forces that are pushing fluid out, which essentially is just the blood pressure in the glomerulus of 60. And in blue, we see the forces that tend to draw or push fluid back in. The colloid osmotic pressure of the blood draws it back in, and the hydrostatic pressure in the capsular space itself tends to push fluid back in. And again, you add all these numbers up, and the net is a net filtration pressure of 10 millimeters of mercury out. All right, so we know that throughout the length of the glomerulus, there is a net pressure that's pushing fluid out. Now, how rapidly does filtrate form? Well, the speed with which filtrate forms is referred to as the glomerular filtration rate. And the glomerular filtration rate, as you can see here, is defined as the amount of filtrate formed per minute. And this is in the two kidneys combined. We generally sum up the activity of both kidneys together and all of these numbers that we're going to talk about. And so the glomerular filtration rate, again, is the amount of filtrate formed per minute. Now, the way we calculate the glomerular filtration rate is we multiply the net filtration pressure, which we just discussed, which is about 10, by something called K sub F. K sub F is known as the filtration coefficient. The filtration coefficient. As you can see in Objective 61, the filtration coefficient is the amount of filtrate produced per minute, so it is a rate, per minute for every 1 millimeter of mercury worth of net filtration pressure. So how much filtrate is going to be formed per minute for every millimeter of mercury of filtration pressure? We multiply that by the net filtration pressure, which is 10, and that's going to give us a unit that is reported in milliliters per minute. In other words, a rate. This filtration coefficient depends on a couple of factors. Excuse me. It will increase as the permeability of the filtration membrane increases, and it will increase as the surface area of the filtration membrane collectively, all added up among the two kidneys, as the surface area increases. Generally speaking, males have a slightly higher surface area than do females, and so males tend to have a slightly higher filtration coefficient. And so what we do to get glomerular filtration rate is we multiply that net filtration pressure, which is 10, times the filtration coefficient, which for males tends to be 12.5 milliliters per millimeter of mercury of force. And in females, it tends to be 10.5, the filtration coefficient. Again, as we said, a little bit less, 10.5. So we multiply that out, we end up with our typical glomerular filtration rates of 125 milliliters per minute for men, 105 milliliters per minute for women. And then again, these are homeostatic averages. Certainly, it does go up and down a little throughout the day. So if you translate what that comes out to, if you multiply this out to what it is per day, you see that you get the kind of numbers that I, in the ballpark of what I referred to earlier, that filtration occurs at a rate of about 180 liters of fluid a day for males, a little bit less, of course, in females, about 150 liters per day in females, but again, still, that's a huge volume of fluid considering that we typically only have 3 liters of blood plasma. So once again, clearly reabsorption is going to be incredibly important to reclaim the vast majority of this volume of fluid that's being filtered by the two kidneys each day. And so what we're seeing here again is another way of saying what I just said. It's about 50 or 60. The amount we filter is about 50 to 60 times the volume of plasma in the body. And so we need to reabsorb about 99% of it. If you think about that, if we do reabsorb 99% of what we filter, that means what's left is about 1%. And so the amount of urine we produce ultimately should be only about 1% of these numbers. Well, 1% for males would be 1.8 liters. 1% for females would be 1.5 liters. And so a typical urine production per day ends up being in this ballpark of 1 to 2 liters of urine excreted per day. Again, that's about, 1% or so of these numbers. Just, again, using ballpark figures. As I mentioned earlier, glomerular filtration rates can go up and down, and certainly they do so, just as body temperature fluctuates slightly around a norm. But then there's also ways for us to alter the filtration rates under certain circumstances, such as exercise and so on. And so we do have a variety of mechanisms for regulating glomerular filtration rates. Just to let you know, if glomerular filtration rate is too high, then generally what happens is we get too much urine produced, of course, because with that excess filtration, the fluid tends to flow more rapidly through the tubules of the nephron, and we don't get a chance to reabsorb as much as we normally would. And so the urine output rises, and of course, if we don't reclaim as much as we need to reclaim by reabsorption, then we're going to lose too much water and too many electrolytes. And so the problem with excess glomerular filtration is, of course, dehydration as we lose too much water in the urine, and electrolyte depletion. If the glomerular filtration rate is too low, then we are not filtering enough materials out of the filtrate to begin with, and so some wastes that normally might get filtered remain in the blood. And then because flow through the tubules is commensurately slow instead of fast, we actually start reabsorbing wastes that we normally wouldn't want to reabsorb. And so this generally results in azotemia, which again, remember, is a higher than normal level of nitrogenous wastes in the bloodstream. So it is problematic either way if the glomerular filtration rate gets too high or too low. And so clearly we have to have mechanisms for regulating it. So how do we regulate glomerular filtration rates? It's a couple of different ways. A moment-to-moment kind of regulation of glomerular filtration rates is essentially performed by adjusting or regulating glomerular blood pressure. Remember from our previous discussion, if glomerular blood pressure increases, we should see an increase in net filtration pressure and therefore an increase in glomerular filtration rate. Conversely, if we decrease glomerular blood pressure, we'll see a decrease in net filtration pressure and therefore a decrease in glomerular filtration rates. So there are a couple of different ways that we do this, that we regulate glomerular blood pressure. And you see those listed here. It's through what we call renal autoregulation, sympathetic control, and hormonal control. So 3 separate mechanisms for specifically affecting glomerular pressure, which is going to affect glomerular filtration rates. All right, we're going to start by discussing renal autoregulation, the first of the 3 mechanisms. Renal autoregulation refers to the ability of the nephrons to regulate their own blood flow and blood pressure in the glomerulus. So this happens without any extrinsic nerve signals or any extrinsic hormonal signals. Now, we will have nerve signals and hormonal signals that will work with renal autoregulation and use some of the same structures. But if you could magically isolate the kidneys from any of the signals coming from the nerve plexus or from any hormones in the blood, it would still have this innate capacity to affect its own blood flow and blood pressure. And this is really the primary mechanism we use to maintain a relatively stable glomerular filtration rate moment to moment, even though systemic blood pressure might rise and fall chaotically sometimes. There are actually 2 ways that the kidneys can do autoregulation. One is called the myogenic mechanism, and that's fairly straightforward. The other is a little more complicated. It's known as tubuloglomerular feedback. So we're going to start with the simpler mechanism known as the myogenic mechanism. Basically, what we see in the myogenic mechanism is that we take advantage, or the kidneys take advantage of some intrinsic properties of smooth muscle, or the smooth muscle tissue that is located in the afferent arteriole. In this particular case, the smooth muscle in the making up the tunica media of the afferent arteriole, this smooth muscle tends to contract when it is stretched. And that's really the basis of this myogenic mechanism. Of course, myogenic means created, genesis, by muscle, the muscle. And so we're talking about something, a mechanism that is created by the smooth muscle in the afferent arteriole. So here's what happens. If arterial blood pressure increases, of course, that might cause an increase in glomerular pressure, which would increase glomerular filtration rate. So how do we prevent that? Well, if systemic blood pressure increases, then the afferent arteriole is going to experience that increase in pressure, and it is going to become slightly stretched. Well, as its walls stretch, the smooth muscle in the tunica media responds to that stretch by contracting. And so what we get is constriction, automatic constriction of the afferent arteriole. So this automatically then reduces blood flow into the glomerulus and prevents the blood pressure from rising within the glomerulus. So as arterial systemic pressure increases, we don't see a commensurate increase in glomerular pressure because of this protective vasoconstriction that the afferent arteriole undergoes. And of course, in the opposite direction, we get the opposite reaction. So if arterial blood pressure falls, well, then the afferent arteriole relaxes. There's less pressure, less stretch on the wall of the arteriole. At that point, the afferent arteriole will dilate and will allow greater flow into the glomerulus so that the flow rate and pressure tend to remain consistent again. And so this is known as the myogenic mechanism. It's an automatic reaction of the afferent arteriole to changes in systemic blood pressure that help dampen those changes at the glomerulus. Now, the tubulo-glomerular feedback mechanism is a bit more complicated, and we need to introduce some new anatomy, some new structures to fully understand the tubulo-glomerular feedback mechanism. First of all, let's describe essentially what's happening, and then we'll introduce some new pieces. In tubulo-glomerular feedback, what's happening is something that's happening in the tubular portion of the nephron is going to feed back onto the glomerulus and affect glomerular filtration rates. And so that's what we're seeing here. It says the glomerulus receives feedback on what's happening downstream in the tubular fluid and adjusts its filtration rate accordingly. So if you think about it, this is a very elegant system, right? The initial step is filtration that happens at the glomerulus. But we're monitoring especially what's happening in the distal convoluted tubule and the loop of Henle. And based on the contents of the distal tubule and the loop of Henle, whether they're too dilute or too concentrated, that information is going to feed back to the glomerulus and affect filtration rates accordingly. So this is a very elegant mechanism for regulating filtration rates, which ultimately then affect the composition of the tubular fluid flowing through the nephron. How does it happen? Well, it happens through something referred to as the juxtaglomerular apparatus. The juxtaglomerular apparatus is a fairly complex structure that is found essentially at the end of the loop of Henle and the beginning of the distal convoluted tubule, just where the loop of Henle ends and the distal convoluted tubule begins. At that point, surprisingly, the tube comes back into very close contact with the glomerulus and the afferent and efferent arterioles. So in most diagrams, the distal convoluted tubule is very far away from the glomerulus. But that's really not accurate. We draw them like that just to make it clear what the different parts of the nephron are. In reality, after we come up out of the ascending limb of the loop of Henle and lead into the distal convoluted tubule, that actually happens very close to the original glomerulus. It's not far away from the glomerulus as you might suspect. And in this region, where the distal tube, the beginning of the distal tubule and the end of the loop, this region where they come back into close contact with the afferent and efferent arterioles near the glomerulus, this region is referred to as the juxtaglomerular apparatus. The juxtaglomerular apparatus has a variety of structures associated with it. So I want to run through some of these structures very quickly, and then we'll put it together to see how they go together, how they work together. So first we have something called the macula densa. The macula densa is a patch of sensory cells that essentially sit on one side of the junction between the loop of Henle and the distal convoluted tubule. All right, so it says here that they're in the nephron loop, but they're really in that transitional zone, again, between the loop of Henle and the distal convoluted tubule. These sensory cells are sensing specifically the concentration of electrolytes, specifically sodium, potassium, and chloride. They're very sensitive to sodium, potassium, and chloride. And so let's think about this. When glomerular filtration rates are high, remember, fluid is flowing faster through the tubes, and so the fluid that reaches these macula densa cells at the end of the loop of Henle are going to contain more sodium and more chloride because they haven't had a chance to be reabsorbed as much. As we said earlier, the faster the rate of flow, the less chance you get to reabsorb. So more sodium chloride in the tubular fluid at this point generally means that the glomerular filtration rate is too high. Well, these macula densa cells are going to sense this higher concentration of sodium chloride, and what they do is they absorb that sodium chloride. They sense it essentially by absorbing it. So the more sodium chloride there is, the more sodium chloride these macula densa cells in the wall of the tubes absorb. Interestingly enough, when these macula densa cells absorb that sodium chloride, they respond by secreting ATP. ATP we're used to thinking of as an energy molecule, but in this case it's also going to serve as a signaling molecule. So just to follow along, if glomerular filtration rate is too high, we have too much sodium and chloride in the tubular fluid, macula densa cells absorb that sodium and chloride at a higher rate, and so they begin to secrete more ATP. That ATP is then going to be picked up by a nearby set of cells called mesangial cells. Mesangial cells. What are mesangial cells? Well, mesangial cells are a separate mass of cells that are located in the space between the afferent arteriole and the distal convoluted tubule. Mesangial cells are in the space between the afferent arteriole and the distal convoluted tubule. So again, this is all part of what we call the juxtaglomerular apparatus, this whole structural system around the afferent arterioles and the tubules. So what do these mesangial cells do that are essentially right next to these macula densa cells, just outside of them, outside the tube? Well, they convert the ATP into adenosine. So essentially they just strip off two of its phosphates, turning it into adenosine. Adenosine acts as a messenger and goes on to then stimulate cells known as granular cells. All right, well, what are granular cells? Here we see yet another new kind of structure. Well, granular cells are also known as juxtaglomerular cells, JG cells, and different books will use either this one term or the other, so you probably should be aware of both, granular or juxtaglomerular cells. These are modified smooth muscle cells in the tunica media of the afferent arterioles and efferent arterioles. Now I said, let me back up, I said they're in the tunica media. They're actually not in the tunica media. They're extensions from the tunica media, and so they're actually found outside the arterioles. It's very unusual to find smooth muscle cells on the outside of an arteriole, but the afferent and efferent arteriole are not like other arterioles, and they have this extension of the tunica media, that extension of smooth muscle tissue from the tunica media that's wrapping around the outside of the arterioles. So realize all these cells are in close contact with each other, right? You've got, because the distal tubule and the end of the loop of Henle come up and pass close to the afferent and efferent arterioles, these macula densa cells, which are in the tubes, are sitting next to the mesangial cells, which are sandwiched in between the tubes and the arterioles, and they stimulate then the granular cells, which are in the arterioles. So they're all very closely organized next to each other. At any rate, the adenosine that's released by these mesangial cells will affect the granular cells, these modified smooth muscles that wrap around the arterioles. Specifically, when adenosine stimulates these granular cells, they respond by becoming excited, causing constriction, specifically of the afferent arteriole. specifically constriction of the afferent arteriole. Well, if the afferent arteriole gets constricted, what's going to happen? We're going to get reduced blood flow, which will then, of course, reduce glomerular pressure, which will reduce glomerular filtration rates. Now, is this homeostatic? It absolutely is, because remember, the original stimulus that got this whole process going was glomerular filtration rate was too high. So through this fairly complicated mechanism, which does not, notice, involve any nerves or any hormones from elsewhere. It's built into the kidney. That's why it's intrinsic. It's autoregulation. But when the glomerular filtration is too high, through this complicated mechanism, we end up with a reduction in glomerular filtration rate. Okay? So the mesangial cells, by the way, which again, the mesangial cells are in between the macula densa cells and the granular cells, they also have some contractile ability, which also might affect the flow through the glomerulus. But that's a minor side function. Really, the main mechanism is as we described it. These granular cells also contain granules of renin, which we are familiar with. Remember, renin is secreted in response to a drop in blood pressure. So this goes back to the blood pressure regulation mechanisms we talked about in our blood vessel chapter. So remember, when systemic blood pressure falls, we said the kidneys release renin in response to that. Well, now we see it's the granular cells of this juxtaglomerular apparatus that release renin in response to that reduction in blood pressure. Remember that the renin then goes on to initiate the renin-angiotensin system of regulating blood volume and pressure. And we will return to this a little bit later, because this ends up being yet another mechanism whereby glomerular filtration rate is affected, but it's done by affecting the entire system's blood pressure. All right, so renal autoregulation, this intrinsic ability of the kidney to regulate its own glomerular filtration rate, is good for moment-to-moment adjustments and homeostatic regulation, but it is not good for in keeping it completely constant, especially in the face of chronic changes. And so chronic changes in blood pressure, basically what happens is the renal autoregulation mechanism just resets to a new set point. And then we're going to need other mechanisms to protect the kidneys if we have dramatic or chronic changes in systemic blood pressure. But it is good for moment-to-moment adjustments around a given set point. All right, and here we just see again, if, as we said earlier, if the glomerular filtration rate is too high, we get that increased absorption of the sodium chloride, which leads to the production of adenosine, etc., etc. So this slide is just repeating what we've already talked about. What I'm trying to get to is a visual of all this, so we can go through it one last time, but with you able to visualize what we're talking about. And here is the visual I was trying to get to. So just to orient ourselves, here is the larger afferent arteriole, and here is the smaller efferent arteriole. So blood's coming into the glomerulus and going out. This is the proximal convoluted tubule leading away from the glomerulus, right? And just follow this little box up here for a moment. Notice the proximal convoluted tubule leads away from the glomerulus, but eventually leads to the loop of Henle, which is going to continue on down this way into the medulla, turn around, come back up. And what I was saying earlier is the ascending limb of the loop of Henle, when it comes back up into the cortex, notice it's not far away from the glomerulus like our previous diagrams would have you believe, but as it comes back up, that end of the ascending limb of the loop of Henle, just where the distal convoluted tubule begins, notice it comes right up back next to the glomerulus and the glomerular capsule, sitting right up against the afferent and efferent arteriole. And then the distal convoluted tubule continues on its way. So the juxtaglomerular apparatus is this region right here where that renal tubule comes back up and sits right next to the two arterioles. So we're focusing right on this region right here. And so this represents the ascending loop of Henle as it's becoming the distal convoluted tubule. And so remember what we said, if glomerular filtration rate is too high, fluid's going to be moving through here too quickly, we'll have too much sodium and chloride in this fluid. That will be absorbed by the macula densa cells, which are these special sensory cells that line the tube right here, and they will secrete ATP. That ATP is going to be picked up by the mesangial cells, which are right here next to the tube, and those mesangial cells are going to metabolize the ATP into adenosine. That adenosine is released onto these granular cells, which are specialized smooth muscle cells that are found on the outside of the afferent arteriole. There are also a few on the efferent arteriole, but too few to be significant. So the granular cells are mostly found on the afferent arteriole. They are going to be stimulated by this adenosine released from the mesangial cells, and they will contract in response. As they contract, they're going to constrict the afferent arteriole. That reduces blood flow into the glomerulus. That reduces blood pressure in the glomerulus, which reduces glomerular filtration rate. So remember, the initial problem was filtration rate was too high, causing too much sodium and chloride here. The reaction or response is to reduce the glomerular filtration rate. And this is all known as the juxtaglomerular apparatus, and it's a form of autoregulation. Now, as I said, this apparatus, parts of it anyway, will also be involved in some neural control of glomerular filtration. Notice you see a sympathetic nerve fiber coming in here, but we're going to talk about that aspect of it later. And we also mentioned that these granular cells are the cells that produce renin when blood pressure is too high. So these granular cells respond to stretch, which is associated with increased blood pressure, by secreting renin, and that's going to be a part of the hormonal regulation of glomerular filtration rate. And again, we're going to talk about that later. One last thing to point out is we said that the mesangial cells were capable of some contraction themselves, and so in addition to ultimately causing the arteriole to get constricted, these mesangial cells are also located scattered throughout the glomerulus, and when they constrict, they can also constrict the capillaries of the glomerulus, which will also reduce flow, pressure, and glomerular filtration rate. So it's a very complicated and elegant way of auto-regulating glomerular filtration rates by the kidneys. And here we see just a sort of concept map of everything we've talked about. Perhaps this view will help you appreciate in a broader sense what we've been talking about. All right, well, we're out of time for today. So next time we're going to pick up with these mechanisms of how we regulate glomerular filtration rates. We have talked about autoregulation, how the kidney regulates its own glomerular filtration rates. What we need to finish up with is the nerve control, how we regulate glomerular filtration rates through nerve control, which is, again, essentially sympathetic control, and then hormonal control. And once we're done with that, we can then finish up the topic of filtration and move on to reabsorption and secretion. So until next time, stay safe.